Thank you, Case, for the generous introduction. Welcome all. It's a pleasure to be here and sharing with you some of the work we're doing at Fresno State in developing new systems for managing vineyards in modern days. We will mostly talk about hyperspectral imagery as a sensing system, and I will introduce you to a series of applications. So we'll start with what is hyperspectral imagery and a little of the fundamentals about the technology, and then we'll take a look at application for grape composition assessment, irrigation scheduling, and detection of viruses. So what is hyperspectral imaging? All of us are familiar with digital cameras nowadays. We have at least one, if not 3 or 5 in our pockets on a daily basis. They take information of the light as a series of numbers from 0 to 255, and then it's with only 3 numbers, we can rebuild 16 million colors, which closely represent what we see with our vision. Now, we can also be familiar with what is multispectral imagery or NDVI, because that's fairly common in viticulture nowadays. And we try to increase the information that we can obtain from multiple wavelengths, multiple different colors, if you want. Also, areas of the light spectrum that we cannot observe with our eyes. The multispectral imagery increase the amount of information, but it's always discrete. It's a series of data points specifically in the physical spectrum of light. Hyperspectral imaging, it takes very little and narrow chunk of lights, but takes hundreds of them. And so we basically can rebuild the full reflectance spectrum of the object under investigation. So if you look at the hyperspectral signature of vegetation, which you see in this plot, with on the x-axis the wavelengths, on the y-axis the reflectance, so how much light is reflected by the object, in this case, a grapevine leaf. We only observe grapevines between 400 and 700 nanometer, which is a place where we look at colors. We look at how green the plant is, how red or yellow. But we lose a very large amount of information that we cannot interact with. And for example, in the near infrared, we have information about cell structure. But also, we have information about water status. All these reflectance dips that you see, it's where the water absorbs in that lights the most. And so the plants reflect less. And this is mostly important in the shortwave infrared. But shortwave infrared also start giving us information about biochemicals, in this case of leaves, but also of grapes. And that's what's used in some of your laboratory machinery. To make sense of all of this information, because again, we are talking of very large amount of data, it's necessary to use very advanced analytical techniques. And in particular, thanks to the advantage in computational biology and agriculture, we can rely on artificial intelligence techniques. So now that I gave this introduction, I just want to showcase a little bit of trials that we have done at Fresno State, starting with assessing grape composition. What we did is why we need a grape composition assessment, because we want to make the best harvesting decision. We want to optimize the logistics and the labor optimization in the busiest time of the year. And also we can open the road to implement variable rate cultural practices, or adapting the management in the vineyard according to the location in the field, if we can map grape composition. And I'm sure there are winemakers in the room, and they always complain that what they have in the tank is not what the viticulture team took samples from, and that's because, of course, we only sample 1% or 1 per thousand of the all grapes in a vineyard. So current methods to monitor ripening are expensive. They need someone to collect grapes, and need someone to crush them, analyze them, and therefore they are time-consuming and inefficient. What we did is that we mounted hyperspectral cameras on the back on a UTV or a mule. And we worked in collaboration with Gallo in a vineyard of Merlot, which was roughly 12.5 hectares and sampled roughly 640 samples on 20 vines, 8 rows, and 4 different dates. We work with shortwave infrared mostly because of the sensitivity to chemical compounds. And this was the first time that this work was done with the shortwave infrared imaging system. And we assessed not only Brix, pH, and TA, but we also did HPLC on the grapes, and we analyzed the full anthocyanin profile. Then the modeling was done through machine learning and the assessment with cross-validation and test sets. So we basically go around in Vineyards at 9, and we put 3,000 watt of halogen light to stimulate the reflectance of the plants. We take imagery of the grapes, and this is a composite of the hyperspectral imaging system. We find all the grapes in the imagery, and we isolate the pure grape pixels. So at that point, what we have is a spectroscopy of the full grapevine population that you can see in the vineyards, and these are Central Valley vines. That's why you can see that canopy on the mule, because we need to lift the vegetation in order to observe the fruit. And if you look at the results in the cross-validation tests, these are Brix, pH, and TA. We roughly are between 7 to 9% of error in assessing these compounds on a simulated new population. If you look at the anthocyanin profile, here you have the total anthocyanins, but also the quality of the anthocyanin, dehydroxylated and trihydroxylated profile. This was only done on the last 2 dates because it's very time-consuming as a laboratory analysis, and we also have roughly 9% of error. Then we went back in a completely different vineyard block and a completely different vineyard, different year to test the system. Well, we just maintained the same varieties. And at this point, I can only show the results on Brix, pH, and TA because I don't have the ones on anthocyanin done yet. But we maintain an error which is roughly 6% on the Brix, 1.9% on the pH, and a larger error on the titratable acidity because 2023 was very different from 2022. And unfortunately, the data that we collected in 2022 were outside the range of the population that we measured in 2023. So how do we solve that problem? By doing more, increasing the number of data and then refitting the model. But what we can do with that? We can have a look at the heterogeneity of grape composition on the plant, berry by berry, as well as you can map grape composition in the field at a spatial scale. Now, how we can use that for assessing grapevine water status? Of course, water is a big problem in California, and because we are under consecutive droughts, there is a lot of competition for water across human activities. We are under regulatory pressure, Sigma, for example, and there is, of course, an environmental concern about making sure that we have water for the next generations, but also water costs money, not only for the cost itself, but also for pumping and distributing. So what we did is that we implemented one of the largest irrigation trials, I think, in the world. There's 12 different irrigation in the same vineyard block that was done with variable rate irrigation systems. And so 12 different treatments, which are different amount of water before veraison and after veraison, and on 4 replicates per treatment itself. The system is an incomplete factorial design, incomplete factorial in a randomized complete block design, totally 16 hectares or 30 acres of vineyards, Cabernet Sauvignon on 1103P, and also that comes from the Central Valley, like the west side. I won't present the results on the irrigation, but I want to present the results on assessing the water status of the vineyard through hyperspectral imagery. So we work with a drone in this case, so we take aerial imagery, and we standardize according to both panels on the ground, but also we tested atmospheric correction that are based on atmospheric parameters. The stress all over the 240 measurements was between moderate to severe, and we separated them according to classes. And we did the prediction through machine learning models from shortwave infrared hyperspectral imagery, which again is very sensitive to water status, but that's the problem that is also sensitive to the water in the atmosphere. And so that's why a lot of work needs to be done in order to bring the results to ground-based information. So those are the results on the classification. What you look now, and you will see a few of them in the next slides, it's what we call a confusion matrix. So on the y-axis, you have the observed data, and on the x-axis, you have the predicted. And those numbers are percent accuracy. So if you see 0.83, that means 83% accurate for that specific class. Or you basically cross the labels on the y-axis with the labels on the x-axis, and you will see how much of that information is predicted in that specific class. So we are able to predict moderate, high water stress with roughly 70% accuracy, and severe water stress with roughly 80% accuracy. There is a confusion between moderate water stress and high water stress. That's because this is only 1 year of data. This is a project that continues for 3 years. And we started in the middle of the summer, so we didn't have a lot of moderate water stress data. And then something that concerns the industry more and more is the ability to detect disease through hyperspectral imagery. And again, we are doing this with one technology and one camera system. What changed is the AI behind the system itself. So how we can detect viruses. Of course, we know that leaf roll and red blotch are one of the most critical diseases in the viticulture in California. It costs roughly hundreds of millions a year. The cost on an acre of vineyards along the 20 years lifespan is estimated to be between $10,000 to $16,000 per acre. And one thing that we wanted to leverage is the fact that diseases affect biochemical and biophysical properties of the leaves, so they change the way they reflect light. So say like, could we use hyperspectral imagery to flag infected vines in the vineyard? And then for that, we mounted what we call the HyperVid project, which it's of course is a riff on the COVID, and it stands for hyperspectral virus identification and detection. It started in 2019, and we have, as of now, through multiple projects, worked on it for 6 years. We started by just making a proof of concept in laboratory settings, like in dark conditions, under specific lighting. Then we moved with still images on the side of the canopy, and finally went to analyze the results taken from aerial imagery. The data acquisition is always the same. We go in the vineyard, we take leaf samples, and we take images. All the leaf samples, every single data point, and this is a very strong difference with respect to other projects that have been done on a similar, with a similar purpose, is that every single data point we have has a PCR analysis behind it. And so as a complete molecular testing. We have roughly 500 images. The data set are these. We have roughly 500 images for the control condition, 700 for the ground-based on the tripod, and roughly 300 for the drone. And then we compared the assessment in an autonomous mode through hyperspectral sensing and AI with the expert assessment through visual estimation in the field. Sorry. For control conditions, we always, again, that was the first proof of concept. We wanted to see if it was also possible for neural networks particularly to differentiate between red blotch, leaf roll, co-infection of leaf roll and red blotch with the not infected. Now, if you ask me, I've been in vineyards for 30 years at this point, and I cannot tell which one is which in these pictures. Instead, so the expert accuracy was roughly 50%. A deep learning neural network system has 80% accuracy in assessing if the vines were infected or not. And the differentiation between leaf roll and red blotch was a little harder. The machine is able to say if it's infected or not, and it generally identified the leaf roll correctly, but the red blotch is generally more challenging to assess, has less evident symptoms. And this was done on multiple dates, and so the symptoms were not always fully expressed. If you look at the red blotch data, because then as the red blotch was harder to predict, we focused on red blotch assessment. And so we went on the ground-based images, so field data with the cameras on tripod, visible near-infrared images, and we acquired imagery over 5 different dates, starting from pre-veraison, when the symptoms are not expressed, all the way to harvest, when the symptoms are at the maximum expression level. And the assessment of the best machine learning model in a simplified way, which only used 18 bands, was roughly 76, 77% for visible symptoms, which is comparable to the expert assessment in the field, where you have 75.3%, or actually slightly higher than that. As an assessment for no visible symptom, at that point, we were under 70%. So we start working in trying to give growers the ability of assess infection before it's actually visible, because that's also a time of the year where you have less to work. Because if you think to monitor every single vine during harvest time, that's not something we can really afford. So this is a total of 700 plants in 5 different measurement dates that were all imaged and PCR tested. And the model performs similarly to the expert using RGB images and higher than them when symptoms are visible. So how to improve the accuracy of non-visible infected plants? Non-visible symptoms are in the plot on the top. So those are reflectance plot of the vegetation under hyperspectral sensing. So on the y-axis, you have the reflectance of the canopies. On the x-axis, you have the wavelengths. We are working the visible and near-infrared, so we are mostly assessing chlorophyll and color. The non-visible symptoms are mostly evident in the near-infrared, which unfortunately we cannot see. So the green line are the not infected plants, and the red lines are the infected plants. While, of course, when the plants start to become red, then you start seeing a higher difference into the visible area of the light spectrum. And if you look at the overall accuracy, we then at that point differentiated the model into 3 different classes. Instead of just saying, a binary classification and say infected or not, we divided into not infected, visibly infected, and non-visibly infected. And so we were increased the accuracy of assessing the non-visibly infected plants all the way up to 77%. And this, I think, is a good strategy that we need to further explore, because first, if you combine the 2 information, like the top plot and the bottom plot, into the same area, you are asking the machine to look at 2 very different things. And instead, if you separate the classes in this way, you are focusing your detection on either the near-infrared, on the short-infrared, or the visible area of the spectrum. And now, just to conclude, I'll present the results of the latest work we have done on this, which is the use of drone imagery for red blotch assessment in the field. So we actually went only one time, because if we want to have very high accuracy, as we need, because again, we need at least 85–90% of accuracy if we want to rogue or eliminate the infection based on a test. And again, the PCR testing is roughly 95% accuracy. That's something to consider as well. We need to go in the time where the symptoms are at the most expressed. So the expert visual assessment, the standard in the industry, is 84% in this particular vineyard, with I cannot really read, 0.92% of true false negative, 76% of true positive, and 8% of plants that are predicted as infected but are not infected, and 24% of not infected that are predicted as infected. The hyperspectral system and the artificial intelligence, it beats the expert, and is roughly 87%. And we have, in general, a slightly higher false positive with the model, which is 10%. The expert was at 8%. But we have a lower false negative, which is more important in assessing the disease. And then we push the hyperspectral sensing more, and then we use radiative transfer model to estimate what are the different compounds in the leaves given their spectroscopy signal. And you can see here a series of box plots where we assess chlorophyll, carotenoid, and anthocyanins, which are affected by the disease, into the infected plant as opposed to the not infected plants, infected in red. And you can see that we can estimate the amount of chlorophyll concentration, the amount of carotenoids, and the amount of anthocyanins. And while chlorophyll and carotenoids, which are part, of course, of the photosynthetical engine, are reduced by the disease, the anthocyanin is increased, and the leaves becomes red. So just to show you a little bit, instead of only presenting results, that behind there is a very straight understanding of how the plants respond to the signal and why we expect the detection to improve or not. So as a summary, all the machine learning models beat the expert assessment. At this point in time, they are not as accurate as a PCR, but we are not very far, because if a PCR is 95% accuracy, we are 87% with imagery. I think we are very close to provide that tool to the industry. At the same time, considering that we use PCR data to assess the disease, you also need to consider we will never be higher than that. So if the benchmark that we train the machines on is 95%, we can expect at most a 90% accuracy on these systems. And another things that we learned is that we need to take the measurement at the maximum symptomatic expression. And right now we're working on wide varieties, and also we're trying to work with understanding the metabolic signal a little better. Just to conclude, of course, I showcase, a part of the work we do in the lab using hyperspectral sensing to enabling precision agriculture to optimize resource and minimize waste. We can try to use advanced sensing systems to enhance the vineyard management. And the idea behind this is not only doing the science, but also to transfer something directly in the hands of the growers that could assist vineyards into their... I finished my time, the bell just rings, but I'm at the end of it. So yes, and so we want to provide something that is available to the growers that they can use directly in their vineyards. And a lot of our work right now is going into simplifying the systems from a full-scale hyperspectral signal to smaller sensors, cheaper sensors that could probably be deployed. I would like to thank you, IVES, for coming to California and for these wonderful days. All the funding sources that have assisted into these works, the American Vineyard Foundation, the CSU Agricultural Research Institute, the CDFA Pierce Disease Glasswing Sharpshooter Board, the CDFA's Specialty Crop Block Grant Program, F3 Future of Food Innovative Initiative, Bronco Wine Company that funds my position at Fresno State. I'd like to thank all the cooperators, which include Bronco, Sansuperi, Gallo, and a number of scientists, colleagues from other universities, Mark Fuchs, Monica Cooper, Brent Sams, Ricardo Pinto de Castro, and Reza Isani. If you want more information about what the lab is doing, you can use this QR code. And thank you to all the people that worked in the lab for these projects. And thank you to you for your attention.